Good morning, everyone, and thank you for being here today. I'm Dr. S.K. Singh, and I'm pleased to welcome you to this presentation. Today, we'll be exploring HVAC requirements in pharmaceutical manufacturing. We'll cover the key aspects of how proper HVAC systems are essential for maintaining product quality and compliance. Over the next few slides, we'll look at the critical design considerations, standards, and best practices. I look forward to sharing valuable insights with you. Let's get started. Let's talk about why HVAC systems are absolutely critical in pharmaceutical manufacturing. First and foremost, they help control airborne contamination, including both particulates and microbes. This is vital because any contamination can compromise the safety and efficacy of the final product. Next, HVAC systems play a key role in maintaining product stability by regulating temperature and relative humidity. Consistent environmental conditions are essential to ensure the quality of sensitive medicines. They also help ensure operator safety by controlling airborne hazards and maintaining a clean, safe working environment. And of course, HVAC systems are essential for maintaining compliance with regulatory standards. These standards are in place to guarantee the safety, quality, and efficacy of pharmaceutical products. Speaking of regulations, oversight comes from major organizations like the World Health Organization, or WHO GMP, the U.S. Food and Drug Administration, or U.S. FDA, and the European Medicines Agency, specifically under EU GMP Annex I. All of these agencies set the strict guidelines that ensure HVAC systems are functioning properly, and that manufacturing environments meet high safety and quality standards. So, in essence, HVAC isn't just about comfort. It's a fundamental part of ensuring that medicines are safe, effective, and compliant with global regulations. Let's take a closer look at basic classification difference. Essentially, this slide highlights how different types of classifications vary from one another. The key point here is to understand that classification isn't a one-size-fits-all approach. Instead, there are distinct categories, each with its own characteristics and applications. When we talk about differences, we're focusing on what makes each type unique and how they are best utilized in various contexts. Recognizing these differences is crucial because it helps us choose the right classification method for the right problem, which ultimately ties back to our overall goal of making accurate and efficient decisions. So, by understanding these fundamental differences, we're better equipped to develop smarter, more effective systems. Now, let's talk about sterile environments first. For critical filling zones, we typically see a Grade A classification, which corresponds to ISO 5 standards. This is the highest level of cleanliness and is essential to minimize contamination during sensitive processes. Surrounding these critical areas, we usually have Grade B environments, which serve as the background to the Grade A zones. These areas still require strict controls, including continuous particle monitoring, to ensure the environment remains within acceptable limits. Moving on to non-sterile environments, these are generally classified as ISO 8, also known as Grade D. While they still need to be clean, the requirements are less stringent compared to sterile zones. Particle monitoring in these areas is typically done periodically rather than continuously, and the microbial limits are also less strict. For example, you might see a tablet compression room classified as ISO 8, which is suitable for non-sterile manufacturing. On the other hand, an aseptic vial filling line would be classified as ISO 5 under a laminar airflow system, highlighting the importance of maintaining the highest cleanliness standards for sterile production. By understanding these classifications, we can better appreciate how environmental control is tailored to the specific needs of each manufacturing process, ultimately ensuring product quality and safety. Let's talk about why having a higher ACH in sterile areas is so important. First, it helps ensure rapid contamination dilution. When we have more air exchanges per hour, any airborne contaminants are quickly dispersed and reduced. This is crucial for maintaining a clean environment. Second, a higher ACH allows for faster recovery after door openings. Every time the door opens, there's a risk of contaminant entry, but with a higher air change rate, the area can return to sterile conditions more quickly. Finally, if we compare air change rates, you'll see that increasing ACH is a key factor in optimizing sterile environments, helping us meet strict cleanliness standards and ensuring safety. So overall, a higher ACH directly contributes to better contamination control and quicker recovery, which are vital in sterile settings. Let's start with sterile environments. For these, the filtration setup includes a pre-filter combined with a fine filter to ensure maximum removal of contaminants. Additionally, the use of mandatory HEPA filters, specifically H13 or H14, is essential to achieve the highest level of air purity. To confirm that these HEPA filters are functioning correctly, we conduct integrity testing using agents like PAO or DOP, which help verify that the filters are leak-free and effective. Moving on to filtration requirements, sterile areas demand rigorous standards because the products are often injectable or highly sensitive. The combination of pre-filter, fine filter, and HEPA filtration creates a robust barrier against particulates and microorganisms. Now, for non-sterile environments, the filtration setup is slightly different. It typically involves a G4 pre-filter and an F7 or F9 fine filter, which are sufficient for reducing dust and larger particles, but don't meet the strict standards needed for sterile areas. In some cases, terminal filters, which are the final filters placed before product packaging, are optional in non-sterile settings, depending on the specific process and contamination risk. For example, in a syrup production area, terminal HEPA filters are generally not required because the risk of contamination is lower. On the other hand, in an injectable filling process, a terminal H14 HEPA filter becomes mandatory to ensure the highest level of sterility and protect the final product. Overall, understanding these filtration requirements helps us maintain the integrity and safety of pharmaceutical products across different environments. Now let's talk about sterile versus non-sterile airflow. In sterile environments, we typically use unidirectional airflow, which means the air moves in a single, consistent direction, either vertically or horizontally. This type of airflow is designed to maintain a clean zone by pushing particles away from critical areas. The velocity of this airflow usually ranges from about 0.36 to 0.54 meters per second, providing just enough force to sweep contaminants away without causing turbulence. This airflow pattern is crucial because it creates a steady, laminar flow, meaning the air moves smoothly in parallel layers. This helps ensure that particles don't get stirred up and remain away from sensitive areas. On the other hand, in non-sterile environments, the airflow is often turbulent. Instead of a smooth, directed flow, the air mixes more chaotically. This mixing ventilation is less about maintaining a sterile environment and more about ensuring general air circulation. The main purpose of these airflow strategies, whether sterile or non-sterile, is to effectively sweep particles away from critical zones. In sterile settings, this is vital for preventing contamination, especially during surgeries or pharmaceutical manufacturing. By understanding and controlling airflow patterns, we can significantly reduce the risk of contamination and maintain safety and quality in the environment. Now, let's take a look at a special case involving cytotoxic sterile products. These require a slightly different approach because they use negative pressure to ensure containment. When we talk about sterile environments here, the minimum pressure we aim for is around 10 to 15 pascals. This helps keep the hazardous materials securely contained and prevents any contamination from escaping. We also follow a strict pressure cascade, which means we have different grades, A, B, C, and D, each with specific pressure levels to maintain safety and control. The idea is that the highest grade, grade A, has the greatest control, and the pressure decreases as you move down through the grades. On the other hand, for non-sterile environments, the differential pressure is a bit lower, around 5 to 10 pascals. The main focus here is dust containment, So maintaining these pressures helps prevent dust from spreading and keeps the environment safe. Overall, whether sterile or non-sterile, controlling pressure differential and following a cascade system is crucial for safety, containment, and preventing cross-contamination in different pharmaceutical processes. Let's talk about temperature and humidity control in our manufacturing environments. When it comes to sterile processes, maintaining precise control is absolutely critical. We need to control for microbial contamination, which is essential to ensure product safety. Additionally, operator comfort, especially during gowning, is important because it affects the overall sterile environment. Process sensitivity is also a key factor. Small variations in temperature or humidity can impact the integrity of the product. In particular, humidity control becomes especially vital when working with lyophilized injectables or during aseptic compounding. These processes are highly sensitive to moisture, and proper humidity levels help preserve the quality and stability of the product. On the non-sterile side, the main goal for controlling temperature and humidity is to ensure product stability throughout storage and handling. For example, tablets are typically stored at around 22 to 27 degrees Celsius, and relative humidity is kept below 50% when dealing with hygroscopic APIs, which tend to absorb moisture from the environment. So, whether we're working in sterile or non-sterile environments, controlling temperature and humidity isn't just about comfort. It's about safeguarding the integrity, safety, and effectiveness of the products we develop and produce. Let's take a closer look at environmental monitoring requirements. These requirements are essential for ensuring that we maintain a safe and compliant environment throughout our operations. First, consistent monitoring helps us track environmental parameters such as air quality, water quality, and soil conditions. By doing so, we can quickly identify any deviations from standards and address them proactively. It's also important to understand that these requirements are not just about compliance. They're about safeguarding public health and protecting our ecosystems. Regular data collection and analysis enable us to make informed decisions and improve our environmental practices over time. Connecting this to our overall theme, effective environmental monitoring is the backbone of sustainable operations and responsible management. It ensures that we're not only meeting regulatory obligations, but also demonstrating our commitment to environmental stewardship. Now, let's take a closer look at the validation requirements for sterile versus non-sterile environments. When we talk about sterile facilities, it's essential to understand that IQ, OQ, and PQ are all mandatory. These are critical steps to ensure that the systems are installed correctly, operate as intended, and consistently produce a sterile environment. In addition, HEPA integrity testing is crucial because the filters are the backbone of maintaining sterility by removing airborne contaminants. We also conduct airflow visualization, often through smoke studies, to verify that airflow patterns are correctly directed and prevent contamination. Recovery time studies are performed to determine how quickly the environment returns to a sterile state after a disturbance. You'll notice that the requalification frequency in sterile facilities is higher, reflecting the need for ongoing assurance of the environment's integrity. Switching gears to non-sterile environments, the requirements are a bit less intensive, but equally important. IQ and OQ are still essential to confirm proper installation and operation, but we don't typically require PQ or the extensive airflow and HEPA testing needed for sterile areas. Instead, focus is on air balancing, ensuring proper airflow distribution, and temperature and relative humidity mapping to maintain consistent environmental conditions. Overall, these validation steps are fundamental to maintaining safe and compliant facilities, but the level of rigor varies depending on whether the environment needs to be sterile or non-sterile. This distinction helps us prioritize our validation efforts and ensure the right controls are in place for each setting. Now, let's discuss the Contamination Control Strategy, or CCS, which is a crucial part of ensuring product quality and patient safety. For sterile facilities, implementing a formal CCS is no longer optional. It's a requirement according to the updated EU GMP Annex 1 from 2022. This means we need to have a structured plan in place to control contamination risks effectively. The focus areas for sterile facilities include several key aspects. First, HVAC risk assessment. This involves evaluating how our heating, ventilation, and air conditioning systems may contribute to contamination risks and ensuring they're properly controlled. Next is filter integrity. Regular testing and validation of filters are essential to make sure they're functioning correctly and preventing contaminants from passing through. Then there's condensate control, managing water that condenses in our facilities, which can be a breeding ground for microbes if not properly handled. Alarm management is also critical. We need robust systems to alert us immediately if any parameters go outside acceptable limits, so we can respond swiftly and prevent contamination events. For non-sterile facilities, the approach is a bit different. They still adopt a risk-based strategy, but the requirements are less complex compared to sterile environments. The core idea remains the same: assess risks carefully, implement controls accordingly, but the scope and strictness are adjusted based on the facility type. Overall, these focus areas are interconnected and fundamental to maintaining a high standard of contamination control, aligning with regulatory expectations, and supporting our commitment to quality. Let's talk about the energy and cost implications of sterile HVAC systems. These systems require higher air change rates, or ACH, to maintain the sterile environment. That means more air is constantly circulated to keep everything sterile and safe. They also use HEPA filters, which are essential for trapping airborne contaminants, but they do add to the energy load. In addition, continuous monitoring is necessary to ensure the environment stays within strict cleanliness parameters, which also consumes energy. Redundancy systems are put in place to ensure reliability, but they inevitably increase energy use as well. All these factors combined mean that sterile HVAC systems typically consume 2 to 3 times more energy compared to non-sterile areas. To put that in perspective, aseptic facility HVAC can account for around 50 to 60% of the total plant energy consumption. So, while these systems are vital for maintaining sterility and safety, they come with significant energy and cost implications that need to be carefully managed within the overall plant operation. Let's take a closer look at the risk comparison. Here, we're essentially evaluating different options or scenarios based on the risks involved. It's important to understand that not all risks are created equal, and this slide helps us visualize where each one stands. By comparing these risks side by side, we can better identify which options are more favorable and which might require additional mitigation strategies. This comparison ties back to our overall goal of making informed, balanced decisions, aiming to maximize benefits while minimizing potential downsides. So, as we review this, keep in mind how understanding these risk levels supports our strategic planning and ultimately helps us choose the best path forward. Now, let's look at non-sterile manufacturing and what it primarily focuses on. It's all about controlling dust, which is essential to prevent contamination and maintain product quality. Product stability is another key point, ensuring that products remain effective and safe throughout their shelf life. Moderate pressure control is also important here, as it helps maintain a controlled environment without the need for the more stringent measures seen in sterile processes. Moving on to the key differences, especially when we compare non-sterile to sterile manufacturing. Sterile manufacturing demands much higher cleanroom grades, meaning the environment must be kept incredibly clean to eliminate any potential contaminants. This is achieved through HEPA filtration, which filters out tiny particles and microbes from the air. Unidirectional airflow is a critical feature as well, ensuring that air flows in a single direction to prevent cross-contamination. Continuous monitoring is essential to detect any deviations immediately, maintaining the integrity of the environment at all times. And finally, strict validation processes are necessary to verify that all these controls are consistently effective. Understanding these distinctions helps us appreciate the level of rigor required for sterile manufacturing compared to non-sterile, and how each approach is tailored to the specific needs of the product being made. For further support, please feel free to contact us. We're here to help with any questions or additional information you might need. And with that, I want to sincerely thank you all for your time and attention today. Your engagement is truly appreciated, and I hope you found this presentation valuable. Remember, we're always available to assist you in implementing or understanding these concepts better. Thanks again, and have a great day.